Welcome to the Rewa Mountains. Today, we're going to be discovering lichen. We get to meet the microbial partners involved in this amazing relationship. We're going to think about how to become like novice lichenologists so that we can differentiate the growth forms. And we're going to think about the relationship that lichens have with us and with other organisms. So hold on for the ride. A lichen is not a single organism. It is microorganisms in relationship with one another. The bulk of the lichen by biomass is fungal. So a fungus shares something in common with us. That is, its cells are eukaryotic. That means that they have a membrane-bound nucleus where the genetic material is stored, and that they have organelles. For example, mitochondria, the powerhouses of the cell. The fungus that is involved in a lichen is a particular type. It's called an ascomycete. Now, this fungus is in a relationship with photobionts or a photobiont. The type of photobiont can vary, and the types of relationships can vary a lot. The photobiont can be an algal cell. Algae, such as Chlorophyceae, so chlorophyta, are green algae. It's also eukaryotic. It has a membrane-bound nucleus, easy to see in this particular electron micrograph, and organellar structure. Here we're seeing the endoplasmic reticulum. But sometimes fungi in a lichen are in a relationship with an algal cell and a cyanobacterial cell, or they're in relationship with a cyanobacterium. Cyanobacteria are not eukaryotes. They don't have a membrane-bound nucleus, and they don't have true organelles. Cyanobacteria are really cool because they do have these structures that are inside of a typical organelle. So they don't have chloroplast, the center of the pigment, chlorophyll, but they do have thylakoids that contain the chlorophyll. So they have some similarities to an algal cell in that way. So photobiont means that these are where the photosynthesis is taking place, that these are primary producer cells. They make food. They are synthesizers of food. So you can see why if you were a fungus, you would want to be in a relationship with a photobiont. Some have referred to lichen as fungi that have discovered agriculture. This is a quote from lichenologist Trevor Goward. Others have referred to lichen as Fred the fungus taking a lichen to Alice the algae. I think all of these oversimplify the relationships that we see in lichens. In fact, in actuality, these relationships can be deeply complex. Sometimes the relationship is between two organisms. Sometimes it's between more than two. Sometimes we see a fungus acting more like a mutualistic partner, that is love-love. Sometimes we could say that, in fact, it's more of a parasitizing, that is the fungus gradually devouring the photobiont. Sometimes we see one lichen infecting another lichen and being parasitic on it. The term cephalodia, when it's referring to lichen, means that you see a three-way association with a primary alga and a secondary cyanobacteria. And this can enable the partners to get what they need from the relationship, for example, fixed nitrogen. But what we can say overarchingly is that the whole is different than the sum of its parts. A fungus that is not found in a lichen looks completely different. So this ascomycete, not in a lichen, looks very different than it does when it's in the form of a lichen. And the same is true of the photobiont partner. So together they make something very new. And in this relationship, we can say that everyone gives a little, everyone gets a little, that maybe the fungus gives some light shield, some moisture, a habitat, a surface, if you will, and then that the photobiont gives fixed carbon. But what we can overarchingly see is these relationships are complicated and dynamic. They are also incredibly colorful. If we were to look at a lichen without pigment, it would be gray, but the fungus and the photobiont give color to this relationship. The melanin that is produced by fungus can give a brown pigment. The chlorophyll that is found in the chloroplasts or within the thylakoids of cyanobacteria is often a very beautiful green. So when moist, the brown melanin may mesh with the green chlorophyll, and we may see an olive color. And while we know that green is common within the chloroplasts of algae and within the thylakoids of cyanobacteria, there's also a lot of other colors. The anthraquinones are orange or red. Some of these pigments are photoprotective. For example, bright yellow eusinic acid that's found within the fungus is effective against short wavelength UV. It's like the fungal shades, protecting from that harmful radiation. This pigmentation is yellow, the eusinic acid, and we're going to talk about it more. And xanthones are also another accessory pigment that are bright yellows. So together we see a rainbow of color. And certainly some of that color derives from the cyanobacteria or algophotobionts. And earlier we were looking at a colorless image. Because of the way that that electron micrograph picture was taken, it made it look colorless. But in fact, this bright pigmentation that is seen in the form of chlorophyll within algae and cyanobacteria. It's so fun for a novice lichenologist to look at distinguishing the 4 different growth forms. And the first 2 growth forms that we want to look at are called Crustos and squamulos. And these are both together the micro lichens, so the small guys. This is a particular picture of a squamulos lichen, but additionally, we can look at Crustos lichens, this very one in the Rewa Mountains. And they are named because they're crusty with cracks that sometimes may seal when wet. They often have a little fringe of fungus around them that is unlichenized. So this is something called the prothallus around the edges. But what's really cool is if you're going to study a crustose lichen, you have to take a bit of the underlying substrate, that is the rock in this case, in order to get it into a form that you can study. It's like inextricably linked with its substrate, the rock there.
Hello, I'm on Evans Gulch outside of Leadville, Colorado, where we just skied up to an old mining homestead. And now I'm appreciating the Crustos lichen that you're seeing on this rock here. And it's beautifully southern exposed. So we're seeing the photobiont being very happy about the access to its energy source. So beautiful spot for Crustos lichen.
Let's look now at the macro lichens, the 2 growth forms, folios and fruticos. The picture here was taken by Christy and I in the Zirkle Wilderness last year. Let's begin with fruticose lichens. Fruticose lichens typically don't have a distinguishable upper and lower surface. Frequently, they have this sort of thread-like or hair-like structure.
Hi everyone, we're up in the snowy mountains where we have discovered a Fruticos lichen. We can tell that this is a Fruticos lichen because it attaches to its substrate in only one location, and then it has this very filamentous growth. It almost looks as though it could be hair, and in fact, I believe that this is witch's hair, or also known as Alectaria sarmentosa. This is Ripple. Ripple went skiing in the fat cat backpack.
And foliose lichen has a really overt upper and lower surface. You can like pick it up like a pancake looking at the upper and lower surface. And it's a very lobular sort of structure.
Hello, we are on Blair Road. We've skied into this particular rock. It's a south-facing rock, and I want to be able to show you a foliose lichen. So this is a particularly beautiful example where you can see the really distinguished undulate edges. And if we were to see a particular kind of foliose lichen, we would see that it could look like a belly button into the earth, or it's sometimes called umbilicate. So if we could take this lichen and look at the ultrastructure of it, it would actually look like a layer cake. And the topmost layer is the upper cortex. Below that layer is where you actually see the photobiont. So that is to say the algal photosynthetic component of the lichen. Below that is what's called the medulla, and then finally the lower cortex. From there, you sometimes see a little rhizome, which is like a little, like the umbilical cord, right, that can attach onto the substrate. So the substrate is the rock, right, here in this case. One thing that's really cool about the cortex of a lichen is that it produces particular cortical chemicals. And some of those, like eosinic acid, is actually inhibitory to other growth. So you might see places where a particular lichen is outcompeting other types of life, whether that be other types of microbial life, or whether that be maybe something like a small tree or a moss or something like that. So maybe up here you're starting to see some of that competitive stuff going on. So foliose lichen, the belly button to the world, if you will, particularly when we think about umbilicate lichens. So we have actually brought with us a tiny microscope that we can use with the cell phone. So we're going to zoom in and take a little bit closer look.
Lichens are incredible environmental indicators. They can help us to monitor the environment with respect to pollution. They are also quite a victim of eco-injustice. That is, they detect pollutants because they absorb far more pollution than other organisms. They're much more efficient at absorbing things like sulfur dioxide, sulfuric and nitric acid, ozone, hydrocarbon, metals. They can detect levels and also direction of movement of these pollutants. So this has been exploited to allow us to very technically detect and calculate an index of atmospheric purity, or IAP for short. This particular map on the left is Montreal, and it shows us the IAP where if we're looking at the city center and the heart of the city here, we notice that these colors of pinkish purple actually indicate lichen deserts, where the lichen has been killed by the environmental pollutants. You'll notice then that radiating away from the city, you begin to see yellows and oranges. Yellow indicates the level of lichens that we would expect to see. And this particular index was looking at lichens and mosses. Fascinating in this image is to look at Montreal. Montreal is a huge park within the city center of Montreal. And you can see that it has recovered expected levels, particularly right at the center of the park there. So lichens can be migrated, in fact, from a native area to an urban area to help to monitor environmental quality measures. So if some measure has been put into place to help to reestablish atmospheric purity, they can be used in a migrated way to allow us to look at effect of those efforts. So lichens, incredible environmental monitors.